100 holes, and I found 3 holes, but I don't know if that was what it really was. 50 RPM, load factor 1.5. So that's easy stuff to understand. And then I made colors, so it's the first 5 is rest, second 6 is exercise, third 7 is recovery. And I put in heart rates from the sheet. I put in VO2s from the sheet, and then VO2 and mLs per kg. I think this one gets calculated. I just took this times 1,000 divided by our kilograms to get those. So again, you guys wouldn't have to show all this stuff. I've got nets. This is just something I created just because I wanted to see what happened. At the bottom, you can see resting VO2. What I did is, if I can do this correctly, resting VO2, I just said, hey, calculate the average of, we said calculate the average of all of these. Column C through 1, 2, 3, 4, 5, so that's what it's doing. And it came out to 0.29. I think that's what we got, maybe. I don't know if that's true. Well, I got 0.28, 0.28, 0.27. We got 3 decimal places, I think.
Yeah.
So let me see if I make this go 3 decimal places. 0.285, maybe?
Yeah, like something like that.
Okay. So that's just taking the average of all these. And then I said, all right, let's see what the steady state is. The steady state, we said, was during exercise, it was going to be the average of 1, 2, 3, 4, the last 4 minutes. So this one says up at the top, the average of C17 through 20. Oops, these C17 through 20. 1.13, that's what I got. And then these other ones, O2 deficit, epoch, you guys didn't have to calculate, but for net oxygen cost, what I did, I said, hey, the net oxygen cost is sum all of these. So it says C15 through the end, all the oxygen uptakes, sum all those, and then subtract 13 times the resting value. And it came, so when I calculated the net oxygen cost, it was 5 liters. I hope you guys got something like that. Maybe if you carried it out to 2 decimal places, maybe 5.04. And you guys get what I did, right? All I did was just add up all the exercise and recovery VO2s, and that subtracted 13 minutes of rest. These other ones, remember I said how you could calculate the deficit? That's the beginning of exercise. The beginning of exercise, the deficit is just, if I add up all the exercise values, and I subtract them from 6 minutes of steady state, and just to do it again, if I did this, here's my beginning exercise, end of exercise, I went like this, I went like that. If I want to calculate the deficit, I don't know if you can see that, it's take 6 minutes of exercise steady state and subtract what you really consume. So when I look at deficit calculation, it says 6 minutes of steady state, 6 times this, and subtract what the person really consumed. And what's left over is 0.739 liters. That was the deficit. The epoch, the end, I said, if you want to calculate this, just add up all the recoveries and subtract 7 minutes of rest. So that's hopefully what this says. It says add up all the recoveries and it subtracts 7 times the resting value, and I get 0.71. So the deficit and the epoch aren't exactly the same. It looks like the deficit was a little bit bigger than the epoch, but they're not exactly the same. So I can share this with you. You can fool around with it if you want. The reason I'm bringing this up is I wanted to show you had questions about plotting and that kind of stuff. So what I did, and you guys can help me if you have better ways to do this, but let's see. This is Excel, so I'm going to just highlight all my O2 values. Oops, right there. And I'm going to say insert I'm going to do a line plot, right? So I'm going to do this and say a simple line plot. So there I go. And here's what I do. I say get rid of this thing. I don't want the title. I'm going to have a caption at the bottom. We had a question, and let me get rid of, I'm going to right click on the format the grid lines. I don't like grid lines on my graph, so I say I don't want a line. Okay, that's just preference. It's up to you guys. Let me move this over a little bit. Okay, then I say I'm going to right click the data points, and it says format the data series. So data series options here. Let's see. Let me see if I do. I can have a line. I can do markers. I'm going to click marker, and I'm going to say my marker options are automatic. I'm going to make them black. Let's see. Maybe it's not going to work. Let's see. fill in. Nah, I can change the markers. Let's just leave the markers blue, I guess. I want to change it to black because I like black, but fill. I don't care about fill. Border on this. Okay, so right now I've got little data points, right? I can do it again. I can right click because we're not supposed to connect the dots, and everybody connected the dots. Format the data series. I'm going to say line. I don't want a line. So now I've got just points. And look at that. I said, okay, that looks like I need axes. And so far it looks like a graph. So here's the tricky part. I click on, make sure this thing is highlighted. And then I'm looking up here. I'm going, all right, layout, formulas, view, design, format. It says add chart element on the left. When I'm at the design, it says add chart element. So I'm going to click on it and say, give me an axis title. Let me get the primary horizontal title. So something just hit right up there, and I'm going to type in time, minutes, and there it shows up at the bottom. And then I'll do the same thing. I'll say add title, axis, primary vertical. You can see it showed up there. I'll type in VO2.
It's like right in front of my face.
Yeah, but it's VO2, and then don't forget the units, so it's going to be liters per minute. And so that shows up. Let me see what it looks like. Yeah, I don't know if this is too close. Let me look at these numbers. Time in minutes.
Can you make it bigger? Like
Yeah, I'm trying to see if, So let's see. When I look at this, I say, all right, so far so good. What I don't like is when I look at this axis, I don't see the line. I see this line. So I'm going to right click here just to see if I can get format. Let's see if I can do it again. Format chart. No, let me try it again. Right about there. It should say format axis somewhere. Somehow I can't. Let me double click, see what that does. No, right click. Format axis. Format axis. It says, let's see, axis options. I want a line. So I want a solid line and I want a black line. So I'm going to put a black line there. Oops. So there's my black line. I'll do the same thing here. Right click on the axis, format axis, axis, options. Where was my line? Tick marks. Did I miss it?
Paint.
I got to do the paint. Okay. Line. I want a solid line. And it's a black line. So now I've got a black line. I've got numbers. If I didn't want, well, if I want tick marks, you can do the same thing. You can right click, you can format the axis, and you can put tick marks somewhere here. There's color, let me see, fill, border. Let me see what that does. Yeah, let me see what that does again. Color, somewhere I thought I saw tick marks, but you get the idea. I'm just playing around here, but I've got my graph, and let's see. At this point, I would just, the easiest way would just be to cut this and paste it in. And then if you want to draw a line, by hand with a ruler, you could kind of estimate where the resting value is and just draw a line right across. You could draw a vertical line here where we start the exercise. At 11, we stop the exercise. And actually, let me right click, format axis, tick marks. Vertical axis, tick marks right there. I want to do major, I don't know, I'm going to inside, outside. Let's try outside and see what happens. I got little tick marks. Kind of funky. They show up in between the minutes. Maybe in between, interval. I like them showing right on the 1, right on the 2.
Right.
So you guys have to figure that out. Time. The other, so you could cut this and paste it and draw all the lines and colors and write stuff in. Otherwise, you could say, I want to insert somewhere here. There's probably a way to insert a text box, and you could put a text box with an arrow. And again, I don't know where these tools are. Insert, home. Because I don't use this. There's tools in here for doing arrows and putting lines. And what I've done in the past is I've found the one that does lines. Who can tell me how to get lines here? There's let's see, insert, charts, table, added charts.
Right click on the graph.
Right click on the graph.
And change chart type.
Yeah, do I want to do chart type? Where?
He wants to put like
Yeah, all I'm
lines
going to
in
find a
it,
tool that
like-
you, there's a tool here somewhere, chart tools,
Like,
I'm looking for insert,
Like the
illustrate.
graph that he drew yesterday, remember how you put like lines in there to
Oh,
like show
like-
like the resting
Page
and
layouts.
then like the O2 deficit and
Something called art, I thought.
Yeah, I just can't,
I think it's on the home.
yeah, I'm looking for clipboard font, alignment, number, style, cells, editing, page layout, colors,
Design?
themes, where?
Add chart element.
Yeah, I don't want that. I'm looking for just a drawing. Oh, right here. I'm looking for a drawing tool. So, so what I did is I said, hey, select this, and let's pretend that that's 1.13. I can, can't do it very good on this table, but I can eyeball that, and I can say, hey, let me do another one, and I'll do one at 5. Again, this is, you know, this could take longer to do by the computer. Let me just copy this. Control C, Control V, made another line. And then I do, I don't know, 11 minutes, put it through the point, and then I could do, I'll copy this one, Control C, Control V. Got another line, I could put, there's my steady state right there, maybe.
Nice, yeah.
And what else do I need? That's about it.
that's it.
You know, and I guess you could do something like this. You could say, hey, turn it around, I guess. I'm just screwing it up. You can see how I would take more care, and I'm not sitting at the right table, but I could say, you know, and then I could just type the text box in, because there's a text box. I could say, this is my EPOC. But here's the problem. So if I do all this stuff on this graph, and I change the size, it screws up where my lines are and stuff. So you got to kind of get the graph as big as you want, and just so the lines don't change. But
Look like they changed with it.
yeah, a little bit. But you get to, and if I wouldn't complain if you just did this by hand, you know, lines by hand. But all I did was I've got an axis with numbers, I've got labels, I've got my data points. Personally, I like them black, but. And this is my preferences. I look at it and say, I don't like grid lines. I like black little data points. I like my data points there, but not a line connecting the dots. You know, in some cases, you might change the range of this, or you might look at this and say, I'm gonna go to my home. This little tool. Let's see if I can do this right. Format the axis. Format axis. Maybe I want the minimum to be 0 and the maximum to be 3 for whatever reason. I don't know. But you can, you know, so you can change the range, you can change tick marks, number. If I wanted my number not to be 2 decimal places, but only 1 decimal place, because it makes more sense to do it that way, maybe, I don't know. But you get it. I'm just playing around and going, now where's that drawing tool again? And I can't even remember where we found it. Where did we find the drawing? Was it design? Format. Yeah, okay. So I would do the same thing. I'd be clicking, going, where the hell's that drawing tool again?
When you're stressing, it doesn't help.
Yeah, this is, so it's the same thing. Yesterday I was fooling around trying to look at, you know, how that looks like. It looks like what we're looking at. There's a deficit, there's the epoch.
So if when you had all those lines in there, you wanted to, and then you have that arrow pointing to the epoch.
Oops.
Which is,
yeah, but now I've screwed up the y
but
axis.
even before it was just pointing to an empty space.
Yeah.
Would you mind if we were able to manually draw in a curve there and shade that area?
Yeah, if you can do that, that's fine. I would personally, if I had to do it, I'd save time by pointing to that area, and then I would just print it in my lab, and then maybe take a pen or a highlighter and just highlight it and say. And if you wanted to draw a line, a smooth line, you know, you can eyeball it if you want, or you can just leave it like that.
Yeah.
But it just connecting the dots doesn't make any sense.
Right. Okay.
So yeah, because really when you connect the dots, I mean that value goes
pretty
up
erratic.
and down. And
Yeah.
you go, it's just really kind of.
Yeah.
So, and I was going to post. See if I post this program. You know, let me get rid of this graph here. Then that means you guys won't calculate any of this stuff. You'll just take it right from mine. I'll post this. I thought I was, yeah, I'll post it, but I wanted to show it to you first. And Amanda and Sarah won't know what to do with this. They won't know what it is, but that's their problem. while I'm thinking about labs, I was looking at labs and comments and stuff. You had the glycolysis. Don't steal from like the internet, even though it looks nice.
It was yours.
Yeah, don't do that. Don't steal mine.
I didn't know how to draw it, and it was
Just draw it with your hands. just take a pencil and go. That's what everybody else did.
You mean like print it out, leave a blank space, and just draw it on there?
Yeah. Or somebody attached it, I think, at the back of the back of their paper, and that was the answer. You just wrote it. So yeah, and I said, I wrote on yours, because I haven't graded them yet, but I wrote on yours, I recommend you draw your own, and then you had a reference, but it wasn't cited anywhere in the text, and I didn't know, when I first saw it, I was riding the bike today, and I thought, was this from this reference? So I went to the website, and I said, no, that's just information about the Wingate.
Yeah.
So if you did take this from somewhere, you'd want to reference it and say,
right.
I don't know, taken from, stolen from. But to me, this is like anybody could cut my slide and paste it. Whereas
I didn't know how to reference your PowerPoint notes either.
Yeah, but I wouldn't even do that. I would just do what everybody else did.
Yeah.
They just drew.
Okay.
And everybody did a good job.
I was desperate at that point.
There was some confusion when I looked at these. I only had 4 on the bike. When I looked, there was confusion about what a figure is and what a table is. A figure is a drawing. A table is a table. And a table has a title at the top, TTT, table title top, and a figure has a caption at the bottom. I have people referring to, they'd say, something's found in table 2. And I looked, there's no table 2. It's a figure 2. Oh, okay, well, they don't know there's a figure in a table. And then they said, something's found in table 1. And table 1 is at the bottom. I said, oh, in lab 0, it says table titles top. You know, so I'll write on yours, C lab 0. If I write C lab 0, C lab 0, C lab 0, that means go to lab 0. And it says, table titles top, figure captions bottom. So that's formatting stuff. With content, I would say there was a little bit of awkward writing, awkward writing. I don't know. People saying things like, in the introduction, here's an introduction from Warren. They said the Wingate test is a way to measure anaerobic power, And then she says, it uses the enzyme adenosine triphosphate. It's like, ATP isn't an enzyme, so I was like, oh, that's kind of sloppy. It uses, I don't know, the Wingate test uses ATP and CP, or uses the phosphagen system, but the enzyme, it's not an enzyme. Somebody referred to, again, I haven't looked at all of them closely enough. So I'm telling you guys things that you'll see later. Statistical analysis. So I didn't see lead-in sentences. Some people didn't put statistical analysis. One person gave all centered headings instead of left justifying the subheadings. No lead-in sentence. Tables on the bottom. Some mistakes like people saying, hey, look at this. It says the peak power was such and such, and then you go, no, that's what you're referring to is the mean power. So I think it's a little bit of proofreading needed to be done.
Can we look at them?
Not now, because I was hoping I could look at hers. I could do them all together, try to be consistent, and then I thought, all right. Normally, what I do is, if you don't do a lead-in sentence for the results, to me that's a 0.25. It's kind of significant. If you don't do axes type labels, I know you had 2 graphs where you made the same mistake on 2 graphs, but I would just ding you maybe a quarter for not having axes. Little things like the wrong date, 0.1. Maybe the purpose. You had purpose in the present tense instead of the past tense. We'll say the purpose was. And it's like, I want to write C lab 0, because it's in lab 0, put the purpose in the past tense. Some of these are 0.1 dings and some are 0.25 dings. I'm struggling with, okay, you guys are urging me to be easy because it's summer and it's the first one. And I go, okay, if I do everything, just be consistent, do everything is a 0.1, then you'll get your lab back and you'll get a 9.5 or a 9 out of 10. And then what will happen in your head, you'll go, oh, hey, I made a shitload of mistakes, but 9 out of 10 is pretty good. I'm satisfied with it. I'm not going to fix anything because I'll keep getting 9s. And so if I ding harder, it gets your attention and you say, oh, God, I better fix these, right? So it's like, which way do I do it?
Well, usually when I get something back like that, then it's easier to like have it in front of me, like what I did, and then I literally will just like use that as a template
Right.
for every other.
And pay attention. So like
Yeah.
when Carlos didn't put page numbers on his,
Oh,
I'm
yeah.
hoping he will say next time, I'm going to put a page number on there. But if I only did him a tenth of a point, he might go, I don't care, it'll be a 9.9 then. Right? But if I ding him a quarter, he's going to say, oh.
Unless
So-
you're a perfectionist.
Okay, so I'm just,
I mean,
I'm trying to balance getting your attention with sort of giving you feedback. The other thing I could do, I'm toying with this, but I don't want to change anything in the syllabus, is say, hey, I'm going to grade this, and if you want to up your grade somehow, rather than redo that one, I've got another lab write-up that I do in the winter term that it's not even in this four-week session that would allow you to do another lab write-up, but I don't want to.
Or what you could have us do is we could go in and correct our mistakes and resubmit it.
I know.
For
that's
like,
what it is.
and not, I mean, obviously not like a huge
Right.
amount of points, but at least it would be enough to like get us to like.
Okay, so
change
what
our
I'll do is
mistakes.
when I look at him this
Experience
weekend, if I decide I'm ornery and I want to really ding him, then I'll do something like that where I'll say, okay, I got your attention. If you want to resubmit for half the difference or whatever, I don't know, then I'll do that. But it's just like I don't want to make you guys do more work, but I don't want to ding you because you haven't gotten feedback yet. So, and the tricky part is it would have been nice to give you this. I didn't look at them early enough, and I should have looked at it so I could give them to you today, and then you're working on your next lab write-up, because I don't know what you guys are doing this weekend, but if you are working on your lab write-up, then you would at least have some feedback. So you won't get feedback until Tuesday. Which
Oh,
is,
that's not fair.
no, but still, you can do most of the lab write-up, and then the little dings and stuff you'll see.
And then just go back and fix it.
Yeah. So the formatting dings are easy to fix. It's the hard dings are. I wrote on Mr. Gonzalez. I was looking at your introduction, and I wrote at the bottom. You said you talk about aerobic, anaerobic, talking about peak, mean power, wind gauge cycling test, and then okay. And I said, okay, so you told me all this stuff, but then I wrote, what is the significance or the importance of measuring anaerobic power? Like you told me kind of what it is, but like, who cares? So the introduction is, what's the significance of it? Carlos said, wind gauge cycling test, blah, blah, blah. He describes it a little bit. It can provide useful information for members of a team so their training can be adapted to suit their abilities, and so that they can eventually reach a certain level of fitness and ability. It can also be used at the start of a training program and after training to see how much improvement has occurred. So that's like the importance of this is here's what anaerobic power is, here's what a wind gauge test is. The importance of doing this is so you could look at training, you could test people and maybe distinguish who's got high power, who's got low power. So that's the hard part for me to sort of, the feedback you get, you're going to get feedback that says, what is the significance? And if that's harder to fix than putting page numbers on. So I'm rambling. But you guys, what I want you to do is when you get the feedback, you'll be able to ask me questions and say, hey, all right. Was I going to do anything else on this stuff? Are we done with this?
I think we pretty much got the main part,
Okay, I think.
which was trying to
pretty
figure out how to
confident.
put names on the axis.
Yeah. Here's Sarah Walker. This is what comes out of the Parvo cart. Oh, see, it won't let me do anything. I don't know why it does this. It just recently, maybe it's a new edition of Excel, but this is what it looks like when it comes out. This is your data sheet that you guys got. So when I put my flash drive into the computer and said, hey, Parvo, send me the data, put it in an Excel spreadsheet, put it on my flash drive, and it did, and this is what it puts. So you can see a lot of useless information, all this stuff that we didn't really care about. And then everything's like carried to 6, 7 decimal places. So what I did when I looked at this, I said, and I can't edit this because protected view, it's because it's a different Excel. I don't want to go through this, but I'm supposed to be able to get to it. And then I just highlight things and say, when I edit it, let me see home, if this was available, that little tool scrunches all the decimal places over so it's readable. So sometimes instead of just, I'll just download it from the Parvo cart and fix this, but all you guys have to work with is just the VO2s and the times. So really it's only 18 values. It's really just 18 values. So it's not really hard for you to type in 18 values in a column. I just wanted to show you what comes out, and it's.
And do we want two to the hundredths digit?
What did I do?
Two significant figures.
Yeah, I don't know what I did on mine.
So we're consistent with it, right?
I did 2 for MLs, 2 for.
Okay, so
Yeah, I guess 2. Again, I'm not big on significant figures because I've done it before, but I'm not anal about that, which you guys don't know. But when I do VO2s in liters per minute, like a 1.12, to me makes sense. MLs per kg per minute, it would probably be okay to be 17.3. And it's like, how do you guys know that? It's body weight in pounds. How many decimal places would you? If I stepped on a scale and I weigh 181.75. Would you guys? Is that okay with you? I mean, you could say 0.75 or 0.8. To me, it wouldn't make any difference. Height in meters should be 1.82. Because if you said 1.8, you could be 1.8. I could be 1.8, but I'm 1.81. You're 1.89. So 2 decimal places. I guess just don't go crazy and put 25 decimal places.
Kilograms you can have.
I won't
60.
I won't ding. Let's just say
height in 1 meter.
All right, let me go to. We're talking about the muscular system today. Boy. And I gave you guys these, right?
Yeah.
I think.
They were on D2L.
Or they're on D2L, sorry. And so again, what I tried to do is what I did before in the nervous system. That the stuff that's review, I'm not going to emphasize and put on exams because it's anatomy phys. So do you have to know how many skeletal muscles? Over 400. No. You know what skeletal muscle is. You know what it does. You know what it's for. So review it. Again, these books start out with anatomy phys kind of stuff, and then they talk about the exercise. So there might be some anatomy phys where I say, yeah, know it just in case. Epimysium, paramysium, endomysium, you guys know that. You know what fascicles are, bundles, okay? You guys have all seen this review stuff. This is a telescoping view of a large muscle going all the way down to myofilaments. What do we look at? If you're interested in epimysium, paramysium, endomysium, I look at that and I say, okay, I've seen it before. Structure, sarcolemma. It says potential exam stuff, and I'll tell you why. Let's see. Potential exam stuff. Sarcolemma, we know, is a muscle membrane. Myofibrils are composed of actin, myosin. I think it's just knowing what a thick and a thin filament are, and what a sarcomere is. So when I say potential exam stuff, is this familiar stuff to you guys? Actin, myosin, and sarcomere? Okay, it really is. Let's see, I'm trying to think of what exercise. It is going to be related to exercise and stretching and muscle length. So I guess it's important to know that thin filament is actin, myosin is a thick filament. The functional contractile unit is called the sarcomere. And a sarcomere is just one Z line to the next Z line. And the picture here says, hey, here's a sarcomere. From Z line to Z line, that's a sarcomere. And what does it have in it? It has a thick filament, that's myosin. It has thin filaments, those are actin. Those are 2 structural proteins. So here's, down at the bottom here, I don't know if you can see this, but there's the myosin with the tails. And it's, the tails are almost touching the actins. The actins are the blues. So I look at that and I say, yes, it looks familiar. This whole A band, H zone, M line, Z line stuff is nice to know, but it's not need to know. In the old days, I would ask you, where's the A band? Where is the I band? Where is the H zone? But again, that's anatomy phys. Anybody know why they call it I band and A band? Okay. I stands for isotropic. A stands for anisotropic. They shine light through it. And when you imagine, and it's hard to do, imagine me shining a light through this stuff down here, or maybe shining it up and I'm looking at it. When I look at it, this stuff's going to be really dark because the light's being blocked, right? So between it, the A band represents the myosins because it's really dark, right? And then the light comes through here, so it's lighter. Oh, that's interesting. This is a little bit lighter because this has to go through the myosin, actin, myosin, actin, myosin, actin. So it's dark. This just has to go through myosin, so it's a little bit lighter. M line is M for the middle. So and the H zone is just the middle portion that is right there. So again, it's nice to know, not need to know. You guys know how this works, right? So here's another telescoping figure with just another sarcomere that says, hey, there's a sarcomere. You got the H zone in the middle, you got the M line in the middle, you got where the thick and the thin filaments are. So again, I don't want to insult your intelligence, but you know how this works, right? The actin slides past, the myosin heads pull the actins. Does that ring a bell? Yeah. Okay. So when you look at this on the bottom, if you guys can see it, I've got these little tails. Let me see if I got a better picture. Here, wow, there's a hell of a picture with more proteins, nebulin and elastin, titin. So I show this only because when I learned it, it was easy like this, and now you look at a exercise phys book is a little higher order, and there's other proteins that are keeping things in place and structurally, and so it's a little more complex. But Z line to Z line, let me see if I got a better picture. Ooh, that's even worse, right? Look at all these. Purpose is to make you appreciate, or hope you appreciate how many different proteins and things are associated with this. If I want a simpler picture. I guess this is as simple as it gets. If these guys, these little heads on these myosins, I'm going to pretend I'm a myosin. This is a myosin right here. These are my two heads right here. So I'm going to connect to actins, and I pull forward, right? So I do this. What's going to happen to the Z lines?
They're going to come closer.
They get closer together, so that makes sense. So the Z lines are going to come closer together. What's going to happen to this H zone right here? It's going to get smaller, right? What's going to happen to the A band? Nothing. Nothing. My A band is just my arm span. I'm just pulling these two sliding things toward me, making the H zone get smaller, but I stay the same, right? So nothing happens there. What happens to my I band, the one that's right here? It's kind of between two sarcomeres. If I do this, if this guy pulls shorter and this guy pulls shorter, what happens to this I band? Does it get shorter?
It's stretched.
It gets, no, I think it gets shorter. I have to think about that one, sorry. Because I'm thinking, let's see, if I pull those together.
Well, I'm thinking like if they're both pulling,
right,
they're going to be pulling this way.
You're thinking like this, right?
Like
so,
if they're both pulling in, like they're going to be pulling that part. I don't know.
I think my sort of first impression is this I band, I'll double check, actually does get smaller. Anyways, I think, and that's kind of stuff you do in an anatomy phys question where somebody might just have a multiple choice question and say, when the myosin pulls the actins, what things shorten and what things don't. I want you guys to appreciate that the myosin's pulling on the actins. All right, and we'll get back to that later just as a review. Review of the structure, microstructure. Sarcoplasm is like cytoplasm, but it's muscle. You guys know we have a sarcoplasmic reticulum that stores calcium. I've got this other stuff that I've seen in anatomy phys, transverse tubules and terminal cisternae. We'll see that in a picture. Mitochondria, we've got mitochondria in muscle. So here's the review picture. You go sarcolemma, cell membrane. Okay, there's transverse tubules. It's called the triad, right? Transverse tubules and the terminal cisternae. Remember that stuff? So I look at sarcoplasmic reticulum. Look at all that reticulum. What's that for? That's for storing calcium. Okay. Mitochondria. All right, I got mitochondria. There's mitochondria that are, they call it subsarcolemmal. It's like right below the cell membrane. There's also mitochondria that are called intermyofibrillar. So there's mitochondria all over the place, depending on what kind of fiber you have. And all right, so we know that stuff. What important things are calcium is stored in the SR, and calcium is going to be important for muscle contraction. Here's another pretty picture. We got myofibrils. We got the actin and myosin filaments, Z line to Z line. I got a bunch of sarcoplasmic reticulum, some mitochondria, the terminal cisternae, and the transverse tubules, that triad, and looks like a muscle fiber. Okay. And so those are pictures we see in anatomy phys books. Neuromuscular junction, where the neuron meets the muscle fiber. There's a motor end plate, a cleft, with a short gap, acetylcholine is released, and it causes an end plate potential. Just to emphasize, when we talked about nerves, we said presynaptic, postsynaptic. I'm going to touch the postsynaptic guy, and the postsynaptic may or may not be polarized, just depends on the input. There's IPSPs and EPSPs that say whether the postsynaptic depolarizes, right? In this case, I've got a neuromuscular, I've got this nerve coming down. When there's an end plate potential, the signal always makes the muscle fiber contract. So it's not like the muscle has an IPSP or an EPSP. The muscle, when I send that signal down the motor neuron, that fiber contracts. And all the other fibers that are associated with that motor neuron. So there might be, this is only one fiber. Maybe, imagine a big picture where this motor neuron is connecting to this fiber, another fiber, another fiber, another. That's the motor unit. So once I send that alpha motor neuron says contract, however many are attached, could be 10, could be 1,000. Remember the innervation ratio is how many muscle fibers are associated with that motor neuron. All of them contract. And we talked, I remember seeing the term innervation ratio, right? That's how many muscle fibers you have per motor neuron. When would you have I may have asked this before, when would you have a very high innervation ratio? Meaning, when would you have a lot of fibers attached to a motor neuron? So here's my motor neuron, and I've got 5 fibers attached. When would I have a case where I have a motor neuron with 1,000? So that's an innervation ratio of 1,000. Here's an innervation ratio of 5. I'm just attached to 5 fibers. What different muscles might have those different characteristics?
Gastrocnemius.
Yeah, be a little more specific. Like what?
Gastrocnemius.
You needed more.
Mhm.
Right?
Gastrocnemius is one.
Okay, why? Why did you pick that one?
Because.
And which one would it have? The 5 or the?
Oh, a lot.
A lot.
Because you need to. It just has basically one purpose.
Okay.
You just need
So I agree with you. I'm thinking, like, to me, it would be like if I have a lot of fibers attached to a motor neuron, it's probably my glutes, because that's gross motor. Once I send a signal down, all those 1,000 fibers contract. This is what I do. I jump. My eyes, my lips, my fingertips, where there's fine motor control, I might want to send a signal where only 5 contract, or 10, or 15. So when I want fine motor coordination, I'll probably have a lower innervation ratio. And that makes sense. And fast twitch, slow twitch too. When I'm on a treadmill and I'm doing a lot of hard work on a max test, remember, I'm recruiting more fast twitch fibers because, boy, I need some help to stay on that treadmill. So this picture, we've seen this anatomy phys and then acetylcholine and just review. Okay, review muscle contraction. You guys remember the sliding filament model. So the Z lines get closer together because actin filaments are sliding over the myosin. There's cross bridges and there's a power stroke. So when I do this as a myosin molecule, that's a power stroke. And so that just moves a little bit, but I'm going to be doing this a lot. So it says, one power stroke shortens the muscle only about 1%, but as long as there's calcium around, I'm going to be doing this and pulling, and I'm just doing 2 myosin heads. There's like a lot. So I'm going to be really, really pulling as long as there's calcium around. And I shorten the Z-line distance. So that's kind of the sliding filament model. Here we go. This is what we were talking about before, potential exam stuff. Let's just call it review. It says sliding filament model. We're looking at H zone is in the middle. When my Z-lines get closer together, this shortens. We said that shortens, right? It's shortening. See right there and there. So the Z-line, the I-band, that does get shorter. So it's instead of getting wider.
Yeah.
So, but it's just, again, that's just review. You look at these and you say, I get it. These myosin heads are going to pull. All right, and they're going to pull a bunch of times. Fine. All right, review, actin-myosin relationship. Actin is a structural protein. Myosin is a structural protein. It has heads and tails. So here's the tail and here's the head. And we're not even going, there's more to this. There's more to this. So this is the simple version. when we look at actin, we've got some regulatory proteins that are troponin and tropomyosin. And I don't know if you guys remember those, but there's a picture coming up. Here's my actin. It looks like two pearl necklaces twisted around. And I say, okay, I've got the actin there, and I've got these little cross-bridge sites where the myosins can attach. Actin is the pearls. I've got a tropomyosin, which is basically, think of tropomyosin as this protein that's just getting in the way. I think of troponin as, hey, troponins are there. They've got a calcium binding site. What they'll do is, if calcium binds to this, it'll pull the tropomyosin out of the way. So one way to simplify it, and it's kind of a goofy way, but I like thinking of tropomyosin as the bad guy. It won't let me contract. So if calcium comes around and attaches to troponin, troponin will pull the bad guy out of the way, and now these sites are exposed, and I can pull with my myosin heads. That makes sense? It's kind of simple. This slide, I look at the slide and I go, I get it. Tropomyosin's in the way. Troponin pulls them out of the way. What's the important thing? Calcium. Man, I need calcium. If calcium's not there, ain't gonna happen. Then the next question you might have is, well, when is calcium there? Calcium's there when there's a nerve, when there's a signal from the motor neuron. The motor neuron somehow, I don't know how this happens, but it somehow causes calcium release from the sarcoplasmic reticulum. So as long as there's a signal that says contract, calcium's being dumped out, calcium's doing its job by attaching, troponin pulls out, so as long as calcium's there, I'm contracting.
So myosin connects or attaches to the cross-bridge binding sites, correct?
Yeah. So if these are available, myosin heads will attach and pull, attach and pull, attach and pull. And they'll keep doing it until tropomyosin gets in the way. And the only way tropomyosin gets in the way is if there's no calcium. And if there's no calcium, troponin can't do its job. And you say, well, when does calcium get removed? Well, calcium gets removed when there's no signal on the alpha motor neuron, and it's taken back up by the sarcoplasmic reticulum. So I think this is a lot of review. I appreciate calcium. That's probably a pretty important thing to have, and it's a highly regulated. It's highly regulated in your body. It's an important thing. If you don't have calcium, you're screwed. Potential exam stuff. This is just another picture of the same stuff. I got myosin tails, myosin heads. They're connected here. One thing it doesn't show up on here, it shows that there's a calcium binding site, but there's also an ATP binding site. So remember, ATP is going to be involved in this. So I look at this whole thing and I say, I get it. This is going to attach and pull if there's calcium around. Got it. I'm going to be doing something. And when this whole thing pulls, and I'm doing this pulling, ATP is going to be broken down. So you go, oh, well, how am I going to get the ATP back? Well, that's that whole bioenergetics thing, glycolysis, Krebs cycle, all that. We're breaking this stuff down when we're contracting. So that's why we talked about bioenergetics early on. And it's a simple cartoon-looking thing, but it does a good job of sort of showing how things happen. Remember I told you guys that I played that science song, and I did. I think I played My Sharona, the one.
No, you talked about it.
He goes, myofibril. And the words to the song, when I listen to it, at one point he says, instead of saying the myosin pull the actin, it says something like the myosin slide past the actin. He says it backwards or something. And it's like, no, I'm attaching and I'm pulling the actins. Anyways, but you guys get this. When you look at it, you won't have to draw it. I got to change this to review, because does this make sense, you guys?
Yeah.
It's review to the extent that if I said it's on an exam, you'd say, oh no, tell us more about it. I need more information. But if I say I'm not going to ask you to draw this on the exam, then you say, yeah, that's review. Probably, right? You guys are more worried about what's on the exam than you are worried about it. Yeah. Do you remember this?
Nice to know, yeah.
Okay. Review. Energy. ATP is required. Got it. There's something. So here's a myosin head, and there's an ATP binding site here. They call it myosin ATPase. This is the enzyme, right? So myosin ATPase is an important thing, partly because, and I may have mentioned this before, when we do a muscle biopsy and pull the muscle out and slice it and stain it, they'll stain for myosin ATPase. So there's different kinds of myosin ATPase, and you can imagine maybe there's a myosin ATPase that's really fast, and that might be in fast twitch fibers. There's a myosin ATPase that's really slow, and maybe that's in slow twitch fibers. And that's kind of, sort of a simple way to look at the importance of myosin ATPase, but it's the enzyme. It's an ACE. Where do we get the ATP? We know this. Phosphocreatine, glycolysis, oxidative phosphorylation, all the metabolism stuff we talked about. So this is where it comes together, where we're using ATP and we need to get some ATP, so that's why it's combined. You guys agree that it says ATP is required for muscle contraction. So if I'm contracting, it means I'm breaking down ATP. If I'm breaking down ATP, I'm making ADP. If I'm making ADP, that should be the signal to turn on all the sources of making ATP. So this is kind of like it says review, but it really, I think it explains why we talked about bioenergetics in the first place, because we're exercise people and we want to contract. Oops. Next slide. Sources of ATP. This is just a picture of what we talked about. So we got the phosphagen system, we got glycolysis, we got oxygen and fatty acids, we got all that stuff, and basically it says, hey, that's where we get our ATP from. So it's just a reminder.
So you said, no, I know you've talked about this before, the production of lots of ADP should signal the production of more enzyme.
It should predict, if I'm filling up with ADP, you should think he must be contracting because he's splitting his ATP to ADP to do this.
Right.
If my ADP levels are up, that should somehow, in a negative feedback way, say to somebody over there, let's get the metabolic pathways going. And what gets the metabolic pathways going is the rate-limiting enzymes like PFK.
So it is.
So ADP is building up. I'm gonna be a signal to turn on or to stimulate PFK, cytochrome oxidase, isocitrate dehydrogenase. I want to stimulate those things. So they're going faster, so I can get the ATP, and it really makes sense in a negative feedback kind of a way. It would be silly to have, I don't know. Maybe it's not. Let's say it's nice to have a signal from exercise that says I'm exercising, so I can get some metabolism going and make some ATP. Okay, potential exam stuff. This says muscle fatigue. So we know what muscle fatigue, well, we tend to sort of conceptualize what muscle fatigue is. It's you can't push the pedals around anymore, or you can't squeeze a dynamometer, or you can't lift the weight. So it's a decrease in the ability to produce force, right? Perform work, push the pedals around. This, and I should have like number 1 here, it says high intensity exercise, number 2, long duration exercise, because when you talk, if somebody said, what causes fatigue, or discuss why do you fatigue? What you guys should say is, what are you talking about? If you're talking about high intensity exercise, the Wingate test, maybe it is lactate, maybe it is protons, maybe it is free radicals, it's pH changes. It makes sense. When you guys are pushing those pedals around as hard as you could, you fatigued, but it's a different kind of fatigue than long duration exercise, where you might have electrolyte imbalances, or maybe you're running a marathon, you're fatiguing because your glycogen levels are depleting. So there's, that's a kind of a trick question. What causes fatigue? You should just answer the question with another question. What kind of fatigue? Short term, long term? It depends. Some of this stuff is iffy. Like you look at long term exercise. I'm running a marathon. Does free radicals, does that cause fatigue, or is it implicated in fatigue? I think they've got it there because there's probably research out there that suggests that it's related. Electrolyte imbalances. Is it? Well, some people have suggested. Glycogen is one of the clear ones. If you run out of glycogen, they've got studies where glycogen levels are depleted and you just can't push. What I want you guys to appreciate from this slide is, so two things. Somebody comes up to you and says, what causes fatigue? And you say, what kind of fatigue are you talking about? And then if they say high intensity, or if they say, whichever one they say, you say, there isn't one single factor that we know of. Because I couldn't tell you from that list of things if there was a single mechanism. I would say, as maybe a wishy-washy kind of professor, I would say during high intensity exercise, some people have implicated lactate in fatigue. I think it's more appropriate to say when you're making lactate, you got lots of hydrogen ions, which is your pH is going down. you might say, well, how does hydrogen ions cause fatigue? Or how is that implicated? I've seen stuff where people said, well, hydrogen ions might interfere with the troponin, tropomyosin, actin-myosin interaction. I don't know how, but so I would be wishy-washy about it and say there's no single mechanism, but these are some things that are proposed. The last one down here, central fatigue, you may not have heard about, but there's a guy down in South Africa, his name is Tim Noakes, and he came up with this hypothesis that it's called the central fatigue hypothesis, and let's see, big picture is people say VO2 max. Let's say you're going to do a VO2 max test. So I put you on the treadmill slowly, and then we increase the speed and grade until you can't go anymore. You know what VO2 does. It goes up, and you can't go up anymore, and you fall off the treadmill because you just can't consume any more oxygen. He's come up with something that says, no, it's not that you can't consume oxygen or you reach something called a VO2 max. He says there's something in your brain that he hasn't identified, but there's a governor. There's a thing in your brain that says, okay, Gary, when you're on the treadmill, every time the treadmill level and speed goes up, we're going to recruit more motor units so you can stay on, recruit more motor units, recruit more motor units, recruit. At some point, this governor says, look, if we recruit any more motor units, this animal is going to it's going to be a catastrophic event. He's going to stroke out or blow up, or the animal's going to die. So the central governor says, don't recruit any more motor units, fall off the treadmill. So there's been this fight between our textbook authors, Powers and Howley, and Tim Noakes, because Noakes says, oh, there is no such thing as VO2 max, and these guys say, no, 99% of the physiologists say there is. You're saying something about some governor that you haven't even identified. But so it's an interesting sort of thing, thinking that maybe it's not just the muscle, maybe there's something in your brain that says, hey, I can't push the pedals around. So just nice to know, not need to know. I don't know if I should say this, but if you ever take a sports nutrition class with me, ever, or a sports nutrition class, they might have a little article or something where they talk about is protein ingestion. Well, you guys know when I'm exercising, carbohydrate ingestion is kind of normal. Gatorade, sort of glucose electrolyte solutions. I want to maintain my sugar, so that's okay. That's fine. Some people have said sugar plus protein is better. Sugar plus amino acids is better than just sugars alone. Related to the central fatigue hypothesis, some people have said, well, if you have amino acids, there are some amino acids that cross the blood-brain barrier that have to do with serotonin and that kind of stuff, and they basically argue that amino acids can maybe be helpful in terms of delaying central fatigue. So it's more nutrition related, but for our purposes, it's just a kind of an interesting concept. Comes up with central fatigue. All right, excitation, contraction, coupling. How does all this stuff work? I depolarize the motor end plate. Right? I'm sending a signal down. It causes muscular contraction. Nerve, you guys know this stuff. This is, I should say, review. The nerve impulse travels down. Calcium binds, changes the tropomyosin location, exposes the active sites, permits the myosin to attach. So the picture is send a signal down. Calcium gets released from this sarcoplasmic reticulum. Calcium attaches to the troponin. Troponin pulls the tropomyosin out of the way, exposes the binding sites. I can now get the cross bridge to move. Easy. It's just a big picture. Relaxation. How do I relax? I get rid of the calcium. Calcium gets taken back up, and then just take it back up. All right. This is from an older edition of the book. And it's from an older edition of the book. It's doing this whole thing where it says when the calcium binds, we get the movement. That's all I want you guys to know. In winter term, we would go through each of these steps, and here's why I'm pausing and hesitating. Because I don't know, when you guys learn this anatomy phys, here's my myosin, and I'm not attached. I'm just sitting here, I'm happy. nerve signal comes down, says calcium you release, calcium binds to troponin. Troponin, moves the tropomyosin out of the way, exposing the sites. Then I attach, right? So the question is, when does ATP get used? When I thought of it, when I was first learning this, here's what I thought happened. I thought I attached, I broke down ATP, and moved, right? And then I released, came back, got another ATP, and it broke down. And did another one. And every time I did this, I broke down ATP. This is saying, no. What happens is, I have ATP attached here. It breaks down to cock my trigger mechanism. Then when there's calcium available, all this does is attaches and pulls. A fresh ATP comes on, breaks down, and then if there's still calcium around, I can attach it. So it's almost like the ATP breakdown pulls the hammer back on the pistol. Does that make sense? It's not probably not important because I'm not going to ask a question like that on an exam, but it's just when I thought of where ATP gets broken down, I thought it was to pull when really it's to get ready to pull. That's kind of what this says. And in the older edition of the book, they talked about a weak binding site and a strong binding site. So you look at this, it says weak binding site. You look down here, it says strong. And to me, instead of saying weak binding site and strong, I tend to think of is it attached or is it not attached? That seems to be the easy way to look at it. But the important thing to appreciate is ATP is broken down. When calcium is available, it'll happen again and again and again and again and again. It keeps on doing it because calcium's around. Remove the calcium and we're done. Potential exam stuff, properties. Here we're getting into fiber types. So this whole next section, and I didn't set my alarm. I got 10:21. You guys want to break till 10:30? Sure. Let's do that before we get into fiber type stuff. And so 10:31 or so. And then can you turn the light on for me?